follows our standardized agenda, which I'll briefly review so that we're all on the can get a compass bearing for the meeting. We would ask that everyone mute your audio when not speaking as a courtesy to everyone on the call. Using your video camera is optional, but keeping them off again while not speaking helps to create a more secure internet connection. So today we will have just a couple of partner announcements to open with. We have Thom Allgaier, who plans on being here to give us an update on spotted lanternfly. We have a couple of general last-minute updates on biological controls. Then we'll talk about the weed-free gravel and aggregate initiative. We'll hear from our terrestrial coordinator, Robert, on miscellaneous projects that he has been working hard on over the summer. Control work, Hypena opulenta, urban forest work, Black River Trail study. And then he and Brittney will kind of co-present on our early detection progress and some notable natives that they've discovered along the way. We'll hear from Sarra from Oswego County Soil and Water Conservation District regarding their Hemlock woolly Adelgid treatments at Independence Park over the past several months. Then we'll hear from our Aquatic Restoration and Resiliency Coordinator, Brittney, who'll give us some updates on the WISP program for this summer, an update from the work that she's been doing in cooperation with Zach, our GIS expert. She'll also be discussing our phase 2 aquatic restoration initiative. And then we'll also hear from Jacob Wojcik about some eDNA project work that's ongoing. And then we'll hear from our education and outreach coordinator, Megan, on events and some interesting work that, Megan has been initiating over the past several months. We will also end with an open dialogue and some wrap-up comments. I'd also like to note that Megan will be placing hyperlinks into the chat box periodically throughout the meeting, so feel free to cut and paste or copy down as you wish. We have a request from Sarra of the Indian River Lakes Conservancy, who wishes to make an announcement. Keep in mind that we'll also have time for open dialogue near the tail end of the meeting. So Sarra, if you are on, please unmute and go ahead and make your announcement. And hearing nothing from Sarra, we'll go on to Emily Sheridan. Emily, if you're on, please unmute and make your announcement.
Rob, thank you. Well, the first announcement is that my name is no longer Emily Sheridan. I recently got married. I fell for Kyle, so I'm changing my last name to Fell. And so if you see some weird emails from Emily Fell, that's just me. And also, I recently had my my contract change, and I'm now working with the Cornell Water Resources Institute, continuing to be in Watertown and work with DEC as the Eastern Great Lakes Watershed Coordinator, but we have this new partnership with Cornell Water Resources Institute that's really exciting and offers some opportunities for more connections with Cornell and with the water resources and other watersheds, working with them like the Hudson and Mohawk. So you know, hopefully that's beneficial to our region. I think there's been retirements in DEC, and we have a new DEC person joining today that I had mentioned to Rob as well. I don't know if Irene is joining, but we do have Irene Mazzocchi retiring from DEC Region 6 towards the end of the month, and we have a new staff on if he'd like to introduce himself, Steve Slewinski. That's all I have.
Super, thank you, Emily, and congratulations. We'll go back and call on Sarra towards the end of the meeting. I don't see her in the participant list.
I'm here.
Oh, is that Sarah?
I'm having. Oh, it's Irene.
Hi, Irene. Did you have an announcement to make?
Oh, I'm just having trouble getting in, but I just wanted to let Em know I'm here.
Super. Glad you could join us, Irene. So Thom Allgaier from the New York State Department of Ag and Markets is with us. He's with the Division of Plant Industry, and Thom would like to provide us with an update on what's happening with spotted lanternfly here in New York. Thom, if you want to unmute, we're ready.
Sure. All right. Thanks, Rob. Good morning, everyone. I wish I had better news to give, but spotted lanternfly has now been confirmed in 13 counties in New York State. I'll go through the list real quick. Well, all 5 New York City boroughs, they call them boroughs instead of counties. You know how New York City is, they got to be different. So all 5 New York City boroughs, Rockland County, Westchester County, Orange County, Tompkins, Broome, Ulster, and also Nassau and Suffolk County. Now, keep in mind that it's not the entire county that is infested, or Richmond County certainly is, but the other counties, usually it's a very small area, like in Tompkins County, where it's a very small, limited area on the campus at Cornell. Same is the case for Ulster County. It's just over the walkway, the walkway over the Hudson, the bridge there, along the rail trail. There's a few sites. So it's a very limited location in some of these counties. So it's not as dire as it seems. The ones that most concern us at the moment are Broome County and the detections in Suffolk County, which are right now limited to Farmingdale, Amityville, and the area right near MacArthur Airport, which is primarily Bohemia. But it's in close proximity to the wine region out there on the east end of Long Island. So it's a great concern to us, but nothing in the SLELO region. So if there is a bit of silver lining in this gloomy SLF report, nothing in the SLELO prism yet. Thank you.
Hey, thanks, Thom. And I think we've discussed this before, but if the need and opportunity arises to put any traps out in our region, we're certainly willing to help out with that.
Yep, I'm trying to coordinate some expanded trapping efforts by the prisons for next year. We're working internally to find out how many traps we got to order. We'll probably have a call with all the prisons and see what everybody's capacity is, because you know, not all the prisons have the same capacity to monitor traps, and we understand that. So, but we definitely appreciate the help and support, and even just the outreach. So, thanks.
Yeah, and I think you'll hear from Megan. We'll hear from Megan later about an effort along the Salmon River that we've initiated over the past 6 to 8 weeks. So thanks for the update, Thom. There we go. So later in the program, we'll hear from Robert Smith, who will be providing everyone with an update on this year's biocontrol efforts regarding our Hypena opulenta releases. However, we did just receive an update on the status of at least 3 other biocontrols. Actually, we received this just yesterday afternoon from Baron Blasi at Cornell. So we wanted to share this with everyone. As a brief update to some other biological controls. The knotweed complex, if you will, there's a psyllid, Aphilera itidori, which is a sap-sucking insect, if you will, native to Japan. As I understand, it's the only federally approved biocontrol for knotweed. It was released in New York early in the summer of last year, 2020. And after monitoring those releases for a year, I'm to understand that this particular biocontrol doesn't really show any promise. It just does not do enough feeding or damage to the plants to cause them to either die back or not reproduce. So at this point, the researchers do not recommend any further distribution of that particular biocontrol for knotweed. Phragmites, currently 2 European stem feeding moths, Arcanera geminipuncta and Arcanera neuricia or neuricia are the most promising agents in the Phragmites biocontrol program. As I understand from Bernd, in the spring of 2019, regulatory agencies in Canada approved field release of these moths, and the technical advisory group in the United States recommended field releases as well. However, according to Bernd Blasi, USDA APHIS has indicated that without some further evidence of high selectivity, a release in the US probably won't be approved. So I understand that there needs to be some more research to make sure that there's not a crossover onto other native plants, and they aren't going to approve it until they see some of that research. Water chestnut, Galarusella burmanica, which is an Asian leaf-feeding beetle, is the focal agent of the water chestnut biocontrol program. Initial studies down at the Cornell quarantine facility using insects originally imported from China, focused on host specificity testing. And a petition for a field release or field releases of Galarusella burmanica is expected to be submitted to the USDA Technical Advisory Group, or TAG, soon. I can't tell you what soon means, but they're working on that petition now. I have personally been to the quarantine lab at Cornell. I've been made privy to the damage that burmanica have on Trappa. And from what I see and from what I understand, the impact is significant, almost completely defoliating the Trappa plants to a point where the plants simply can't reproduce. So if and when that biocontrol is approved, I'm of the opinion that this particular biocontrol could be a game changer in our efforts to suppress water chestnuts. So fingers crossed on the research that's ongoing, and hopefully the acceptance of the TAG petition to do field releases, hopefully within the coming years. I also wanted to give everyone an update on the weed-free aggregates pilot project that we're trying to get underway here in the region. You may recall this past year, we participated in an endeavor that would pilot this program, allowing for weed-free aggregates. Katie Malinowski was our strongest proponent and took a lead role in this effort. The program provides quarries with tools to screen aggregates for seeds and plant materials, and to provide those aggregates at a premium. And it would also establish trained independent inspectors to certify the material. It does require a memorandum of understanding with an oversight agency, and currently there are 14 or more soil and water conservation districts interested in providing inspections. So it's a popular program in the Western states. There's strong interest to do it in our region and here in New York. And it's a program that's been primarily sponsored through NASMA. So this past year, we submitted a proposal which was not funded. This fall, Ag and Markets has prepared a fresh proposal, which we hope will be funded in 2022. And the anticipated outcome, again, is to reduce the amount of contaminated aggregates from being moved about the region. So we'll keep you posted as the process develops, and again, hopefully with favorable results. This photo here, I know it's a terrible photo, but I just had to throw this one in. This was a photo that was recently taken in our region. It was one of those, drive by, hold up the phone and snap a photo opportunity. So I apologize for the quality. And yeah, I know we're not supposed to be messing with our phones when driving, but I just had to try to capture this. So what this shows, if you look carefully, is on both sides of the excavator, there's knotweed, and in the far side of the field, there's knotweed. And to the left of the sign on the side of the road, there's Phragmites. So we have this corner lot that's dominated with Phragmites and knotweed. And so the excavator cuts a path right through the knotweed to gain access into the interior of the field. And so I guess I wanted to show this just to document that this kind of thing still takes place. We've reached out to heavy equipment contractors in the past with help from our DOT rep, Becky Miller, our former representative from DOT. We presented contractors with clean equipment protocols, how-tos, etc. But ultimately, this type of behavior continues. So we need to, I think, consider alternate ways to try to reach out to contractors and heavy equipment operators to do some education and try to change this type of behavior. I'm sure the excavator will be contaminated and be moved to some other region. And what happened to the spoils of this clearing, I don't know. I don't know if it's still on site or if it's been moved, but just wanted to throw that one in. So this brings us to our Terrestrial Restoration and Resiliency Coordinators report. Robert Smith is with us. He has been very busy with various projects and is going to provide us with a fairly detailed update on his work over the past several months. So Robert, feel free to unmute and begin your report.
All right, thanks, Rob. Good morning, everyone. Going to start off talking about the 2021 control work. Our contractor, Miller's Turf, has completed their control work for the year, and we asked them to work on 6 invasive species that you see listed here, starting with the giant hogweed in May. So that's where I'm going to start. I'm going to start talking about the giant hogweed. There were 48 giant hogweed sites that SLELO Prism managed as part of the New York State DEC giant hogweed control program. There was no germination in 40 of these sites. The giant hogweed was removed mechanically at one site using the root cut method. Herbicide was applied to 6 of the sites, and one site we had no permission to treat, so there's nothing we could do with it. Other invasive species managed this year were the swallowwort, which was managed at 77 sites located on 13 PCAs for a total of 69.5 acres managed. Japanese knotweed was managed at 7 sites located on 4 PCAs, and that was about half an acre that we managed. Phragmites was managed at 5 sites on 3 PCAs for about 3/4 of an acre being managed. Oriental bittersweet was managed at 3 sites located on 2 PCAs for a total of 6 acres managed, which this is a big change from last year because I'm sure many of you know we nominated this last year as a new tier species. So we're heavy on it already, and we're managing 6 acres to start with. And I found some more for them to work on next year. So that number will probably increase next year. And
Robert,
yeah,
the swallowwort was 78.9 or 69?
69.
Okay.
Oh, maybe that's my notes. It's like there. Yeah, I'm going to have
right around 70 acres.
Yes.
Super.
Yes.
Thank you.
Yes. Yellow iris was managed at 1 site, real small patch. So we're just talking about a tiny little 1,000th of an acre. And, that was it. Okay. Oops, getting ahead of myself. All right, it's supposed to back up. Back up. There we go. Very good. All right, this year we completed another biocontrol release of Hypena opulenta. As a review, Hypena opulenta is a moth native to the Ukraine that feeds exclusively on pale and black swallowwort in its larval form. The adults have a dull light brown forewings with a dark band in the middle and hind wings that are pale orange. The larvae starts out white, but later becomes green with black spots and a yellow head. The larvae go through 5 instars or life stages. They pupate and then they emerge as adults that are very short-lived. They only live like 2-3 weeks on average. And these adults spend most of their time looking for a mate, mating, or laying eggs. And that's pretty much, the end for their life cycle. All right. The adults this year were placed in 4 cages on June 1st. 2 of those cages were at Whaley State Park and 2 were on Grenadier Island. There was 20 males and 20 females placed in each cage on Grenadier Island. But we didn't have enough to do that at Whaley, so one of the cages we did 20 males and 20 females, while the other cage we did 10 males and 10 females.
Yeah, Robert, I'm not sure why the slides aren't advancing, but maybe I can hit them from here for you.
Okay, I appreciate it. All right. Once we had the adults in the cage, these cages were monitored for a new generation of Hypena larvae and levels of defoliation of the swallowwort in each of the cages. Last year, this process occurred in 4 weeks. And we released the larvae when they exhausted the supply of swallowwort that was in the cage. This year, the number of larvae produced was very low. And this resulted in a low amount of defoliation. We're talking like less than 5%. We were concerned that if we released them from the cage at that point, they would just basically fly off in different directions and never to see another Hypena again and never to produce a new generation. We knew that Hypena opulenta normally produced 2 generations per year, so we decided to leave them in the cage to produce a 2nd generation. We had never done this before, but we thought it would improve the chances that mating would occur. As you see in the graph, this paid off at Grenadier Island with defoliation in cage 1 at 20% and cage 2 at 100%. And this was after 10 weeks because we had to, take that much longer for them to mature into adults, produce a new generation. So 10 weeks, but I believe it was worth it. Unfortunately, the cages at Whaley State Park had even less Hypena there than Grenadier, and they just didn't produce a 2nd generation. All 4 cages were removed on August 16th. We intend to set up cages again at Whaley State Park and Grenadier Island next year, with the hopes that with continued releases, we will it'll result in a long-term establishment of Hypena in these locations. All right, the Urban Forest Sustainability Initiative. This initiative promotes resilience to invasive pest pathogens and climate change through increased tree species diversity, planting climate adaptable trees, planting native trees, pest management, and right tree, right place. This program offers a sustainability guide, a presentation, and up to $5,000 reimbursement for the purchase of non-invasive tree species. The only requirements are that members involved with the city's tree program attend their urban forest sustainability presentation, and that they provide us a receipt showing that they purchased non-invasive tree species, which we define as those not listed on IMAP as invasive species. The villages that participated this year were Messina, which received a presentation on June 3rd. And all of these places are currently ordering trees. So they're planning on a fall planting. Canton received a presentation on October 5th with plans for a fall planting. And Pulaski a presentation on April 5th, and they also plan a fall planting. Pulaski was a little different. They're a smaller community. They weren't on our target list of communities, but since they expressed interest, we were able to offer $2,000 in discretionary funds to reimburse them for their tree purchases. Exact dates for all fall plantings are still to be determined, and I will update you on these plantings during our winter partner meeting. All right, as a refresher to those who attended past meetings and as an introduction for those who did not attend, I'll start with some background about the Black River Trail feasibility study. The Black River Trail is a trail that goes from the village of Black River near Fort Drum to Watertown. It's a very busy trail. In 2019, they estimated there was 104,000 visits to the trail. The area of the trail that we were concerned with was a 3.5 mile segment that is adjacent to the river. There is an abundance of several known invasive species along this part of the trail, and that's really why we were drawn to doing this study. Invasive species include Phragmites, swallowwort, Oriental bittersweet, buckthorn, honeysuckle. The objective of the study was to determine the feasibility of invasive species removal, control, and restoration work, and to create a feasibility report that detailed the results of our study. Our methods for the study involved dividing the trail into 29 compartments and recording such data as tree abundance and canopy cover, herbaceous understory plant abundance and cover, invasive plant abundance and cover, and location of culverts, tributaries, and any other, man-made objects. The results of the survey can now be found in the newly completed Black River Trail feasibility study report. This report lists over 200 plant species found along the Black River Trail. It also provides many statistics like the number of species in each compartment, number of woody and non-woody species, native species and invasive species. There's also, in the appendix, there's a really nice breakdown of the what invasives and not our native species, woody versus herbaceous for each and every single compartment, along with a listing of every species that was found along the entire corridor. Prioritization scores using the system that I outlined in the spring partner meeting are also provided. Costs for conducting invasive species management and restoration work were also estimated for 5 compartment groups that we created based on those priority rankings. We concluded that the invasive species management and restoration is feasible on this trail, but only if available resources are designated according to compartment prioritization scores. We recommended that removal efforts use mechanical manual methods to limit the amount of potential chemical exposure to the public. And we also recommended that native plant species that we found on the trail during the survey those are the species that should be used in any restoration work. All right, we just finished up our summer/early fall field surveys last week. We surveyed around 160 highly probable areas at 11 priority conservation areas, which you can see listed all here. And if you look, you'll see at each PCA whether there was an aquatic survey, a terrestrial survey, or both. So A, B, and they're A, T, and B. Aquatic surveys were completed at 9 of these PCAs, and terrestrial surveys were completed at 6 of these PCAs. We found many tier 3 and 4 species, as we knew we would, such as swallowwort, Phragmites, Japanese knotweed, Eurasian water milfoil, the European frogbit, and many others were found at these PCAs. And we visited a couple of our giant hogweed sites as well that we already knew about, and those were a tier 2 species. But we did not find any tier 1 species, which is a good thing. So better luck, or hopefully we have that kind of continued luck for years to come, but you never know. We are currently reviewing all the data that we inputted into IMAP Mobile Advanced and SAS Pro, and we'll be creating a report that will include what we found at all the PCAs that we surveyed this year. Notable natives. During our early detection surveys, we also see many native plants species when we are looking for invasive species. And these are the species that we are protecting. And we have been recording these native species this year when we see them, to get a better understanding of what species are adjacent to the invasive species that we're looking for. Sometimes some of these native species are rare, and sometimes they're just species that we didn't expect to find. For today's presentation, I have selected 2 terrestrial native species that we found this year that I'll talk about, and there is one a native aquatic species that Brittney will talk about after I'm done talking about the terrestrial species. The first of these species is rock elm, or Ulmus tomasii. It's a threatened species in New York State. It's most common in the upper Mississippi Valley and lower Great Lakes regions. Although its native range includes portions of New Hampshire, Vermont, New York, extreme southern Quebec, and that areas just west into Ontario. The distinguishing characteristic of this elm, really the easiest way to tell it from other elms, is a thick, corky, ridged or winged branches, which you can see pretty clearly in these pictures. The habitat preferences for this species include rocky ridges and summits, rocky outcrops, cracks in and thin soils over bedrock and woodlands. For our area, that would include the Alvar. It is susceptible to Dutch elm disease, like all other elm trees, and so there's been a significant loss in the population of this elm species, like the others. The seeds and buds are useful as a food source for wildlife such as deer, rabbit, squirrels, and a variety of birds. The wood is actually stronger than any other commercial elm, and this has caused it to be drastically overcut in many localities. It also has a bending quality to it, so it makes it good for bent parts of furniture, crates, and containers, and as a base for a veneer. The next notable native I'd like to talk about isn't rare at all. In fact, it's considered quite common. That's pale their jewelweed, also known as touch-me-not. It's, like I said, common. It's about 2 to 6 feet in height. You know, leaves there, they're 5 inches long by 3 inches wide. They're hairless. They're rather oval egg shaped. The flowers are yellow, and what we usually see in this area is a spotted jewelweed, and they have orange flowers. And just to backtrack a little bit, as I was saying about it being a common species in New York, yes and no on that. It's regionally, it can be quite rare. And in fact, last year we did not see pale jewelweed in any of the PCAs we visited last year. This year we got super lucky and we located it at 2 sites. So that's why I'm including as notable native, not because of its rarity to the state, but really because it's kind of a treat to see it up in the North Country. All right, and back to the fruit. The fruit's kind of interesting because when it's mature, it explodes with just the slightest touch. And in fact, that's why it's also known as touch-me-not. This species is a facultative wetland plant, so it prefers wetter habitats like floodplain forest or seepage areas, swamps, marshes, fens, stream banks. This species also has a long history of use amongst Native American medicine, and its sap, or the sap from its stems and leaves, can be used to relieve itching and pain from ailments, including hives, poison ivy, and stinging nettles. Different parts of this plant also attract a variety of wildlife as a food source. Birds like hummingbirds, or hummingbirds specifically, and insects like bumblebees and butterflies are attracted to this flower for the nectar and pollen. And if you look in the picture, the most left picture that you see, so are these hoverflies, which Brittney went through the trouble of figuring out what this was when she took the picture. They all can be identified by their very distinctive long snouts. Moving on to other wildlife uses, rough grouses and white-footed mice like to eat the seeds. And deer and caterpillars such as white striped black moth and pink legged tiger moths like to eat the foliage. So, and there's so many on here. We could have hours there on these notable natives, but I'll just highlight 2 today. Brittney's going to now talk to you about the mosquito fern.
Thanks, Robert. Good morning, everybody. So I hope that you're all enjoying our notable native highlights, just to kind of give you an idea of what we're seeing in the field. And so I'm going to talk about the mosquito fern. And if you look at the photos of this species, you can see that it is a pretty small fern, actually one of the smallest ferns in the world. And it's just a pretty cool encounter. It's a free floating species that forms these like dense mats. So when you do find them, it's typically abundant. And although it's not technically rare in New York State, it is considered to be an exploitably vulnerable species. Interestingly enough, the name mosquito fern actually came from the myth or conversations that if you had an abundance or a dense mat of mosquito ferns on your water, that mosquitoes couldn't actually lay eggs in the water. So you're reducing, they were reducing mosquito populations. So just as a native species, again, it is actually slightly different than some ferns, that it mostly reproduces through fragmentation, but like other ferns, it can also reproduce through spores. And it is a good habitat for both micro and macro invertebrates in the water, a good food source for multiple different species. And something that is kind of interesting about the Azolla species in general is that it's considered to be a really good nitrogen fixer. And in some locations where Azolla species are prevalent, they actually use it for a natural fertilizer in agriculture. Even talking about like rice paddies, where it's actually considered to release 3 times the rate of legumes in nitrogen. So super interesting. It's a cool species that is not something that we see very frequently. I think we only found it in 2 locations so far. But yeah, we just wanted to share that with you. So thanks for listening, and back to you, Robert.
Well, that concludes my portion, actually. So move on to the discussion about the Hemlock woolly Adelgid treatments at Independence Park.
So thank you both, Robert and Brittney, for those reports. And I especially like the notable natives, because, we talk often about invasives and control work, and I don't think we talk enough about why we're doing that, and, contributing to improving biological diversity, while at the same time noting those natives, I think is really interesting and very beneficial. So next, I'd like to introduce Sarra Learned from the Oswego County Soil and Water Conservation District. You may recall our early detection of Hemlock woolly Adelgid last winter, early spring in Oswego County. And Sarra graduated from SUNY ESF last spring with a degree in conservation biology. She worked for Oswego County Soil and Water Conservation District as an intern on their water chestnut program for 2 summers. And she's been working at the district as a conservation aide since this past spring, where she focuses primarily on invasive species such as water chestnut, giant hogweed, and now the Hemlock woolly Adelgid. So welcome, Sarra. You are all set to unmute and begin.
Good morning. As Rob mentioned, my name is Sarra. I'm filling in for Joe today. He's currently presenting at the NYACD conference. He was the lead on this project, so I'll be giving some highlights of the project, but any detailed questions may need to be directed to Joe, and his information will be provided on the last slide. So as many of you know, SLELO confirmed Hemlock woolly Adelgid in Oswego County in February of 2021. The Adelgid was found on 3 county properties, with 2 of these being parks open to the public. It was decided that the 2 park properties were priority areas for treatment in 2021. A survey was needed first, and SLELO organized an event to determine the extent of the infestation. During the survey, approximately 30% of the stems showed visible signs of Hemlock willi Adelgid on the lower branches, suggesting that the infestation was well established. And in this picture to the right, you can see the white cottony masses that the Adelgid produces, and this is what we looked for to ensure infestation. And then the next slide, please. So between the two public parks, Hemlock was found to occupy about 24 acres at Independence Park and an additional 2 acres at Camp Hollis. The photo on the right shows Hemlocks towering over a trail at Independence Park. And if the Hemlock willi Adelgid is left uncontrolled in these areas, it will lead to a safety and liability issue as decline progresses. And then this is followed by eventual mortality. Next slide, please. So the best method of treatment was found to be a basal spray consisting of a tank mix of 2 pesticides. And these pesticides are both listed in the second bullet point on the screen.
You love
mixture
mixing
provides-
pesticides. That sounds great.
I'm sorry. So our mixture provides immediate mortality of the Adelgid with residual control for up to 5 years. When applied by an applicator, this costs about $2.50 per inch of tree diameter. And when using this mix, the label only allows for 170 inches of tree diameter to be treated per acre per year. That makes this a multi-season control project. And using the diameter restriction, the first treatment was estimated to cost approximately $11,000. So throughout the year, Joe approached Oswego County officials regarding authorization to coordinate a treatment and for potential funding. It was decided that the county, along with our soil and water district, would each pay half of the application cost. Our Oswego office arranged for Onondaga County Soil and Water to conduct the treatment since they have a certified applicator on staff and we do not. Next slide, please. So after authorization, there are only 2 windows for effective treatment, that is in the spring and in the fall. Layout began in September, and during this phase, we found that a wetland permit was needed for much of the project. So our original acreage and number of trees to be treated this year in 2021 was reduced a little bit. Trees were selected in March for control based on their diameter, height or crown position, health, and proximity to a trail or parking lot. Each tree was measured to ensure the diameter restriction was followed, and approximately 100 trees were flagged and treated just last week on October 13th. Next slide, please. Pictured on the right is Teresa Link, and she is the applicator from Onondaga County Soil and Water. The pesticide is applied to the first 4 to 5 feet of the trunk with a backpack sprayer until the product begins to run off. Both of our sites were treated in 1 day and cost approximately $3,300 in total. Since treatment took place in public parks, the trails at these sites were closed down with signage for at least 12 hours in accordance with the product label. Next slide, please. And this slide shows examples of what the trees look like after treatment. The photo on the left was taken just 1 hour after application, and then the photo on the right was taken the very next day. The white residue you see on the bark on the picture to the right will remain until it is washed away by a rain event. Next slide, please. So now looking into the future, a wetland permit with the New York State DEC has been applied for, but it may take months to obtain. So we weren't able to use that for this fall treatment. Once this permit is approved, we will treat the remaining acreage we were not able to get to this year. And this is expected to take place next year in 2022, hopefully by the spring, if not then by the fall. The permit will allow application within the wetland and the 100-foot buffer zone. And we do not plan on treating in wet areas, but the buffer zone does contain a lot of hemlock trees in dry land conditions and in close proximity to trails, which is one of our priorities. Beyond 2022, we will organize small treatments until all of the priority trees have been treated. And now on behalf of the district, I would like to say thank you to SLELO, Oswego County Legislator, Oswego County Youth Bureau, and the Onondaga County Soil and Water Conservation District for all of your cooperation on this project. Next slide, please. So now that's all I have. Thank you very much. And again, any detailed questions may need to be directed towards Joe, but I can try my best to answer any questions you may have today. Thank you.
So thank you, Sarah. And I think that, there's been some questions coming up during different presentations through the chat box. Perhaps what we should do is, if you could all hang on to those questions at the end of our meeting, we can have an open dialogue, kind of like a panel discussion, if you will, with all of our team today to try and answer any questions rather than skipping around back and forth. So our Aquatic Restoration and Resiliency Coordinator, Brittney Rogers, has also been very busy with implementing programs and projects. So we'll hear from Brittney now. You want to unmute? I'll give you approval there. And you're all set. Should be all set, Brittney. Brittney, we don't hear you.
Sorry, everyone. Every time I try to start my video, my Zoom actually freezes and restarts. So I apologize. You don't get to connect with me via video today. But hopefully, Rob, if you can just confirm that you can hear me, hopefully my Zoom is not fully frozen.
Yes, we can hear you loud and clear.
Okay, perfect. Thank you so much, everyone. Sorry again for that short delay. Technology is always fun. Yeah, so as Rob mentioned, I've been super busy with a bunch of different programs and different events. So just wanted to share a little bit of this with you. And so first I wanted to start by talking about the Watercraft Inspection Steward Program. So this year, the first three lines or the first three numbers you see are the total surveys that were collected this year. So we collected 8,500 watercraft inspection surveys. And we added 2 new surveys, and one of those is called Walk Up. And this is basically if anyone's at a launch where a steward is stationed and they just, without a boat or fishing equipment, walk up to the steward to ask questions and learn about invasives. We just kind of gathered a little bit more information because we realized how many people the stewards were talking to without boats, but we weren't actually capturing that information well. So that was a good increase. And then we also included an angler survey. And so we conducted 160 surveys of anglers and their fishing equipment and gear. We talked to 21,000 people this year, almost 22,000, which was about 6,000 less than last year. So I think with COVID kind of changing a little bit this year, we had less people at the launches because many people were back to work and weren't as free as before. And then also some of the weekends, the weather just wasn't great and our stewards just didn't have as many boaters at locations on top of low water levels along Eastern Lake Ontario, which were some of our busiest launches last year. The total inspections is actually because we often, if there's a group of boats in one group, we'll survey all of them together. And so you'll see a higher number of actual watercraft inspections than total number of surveys. And for the most part, we had a higher number of people launching versus retrieving, although most of our interceptions of invasive species were actually on boats that were coming out of the water. So this was good because being in the SLELO Prism, we are connected to the Great Lakes, which do have the highest concentration of aquatic invasive species. And that gives us the opportunity to actually try to reduce that spread of invasive species from the Great Lakes into inland water bodies like the Finger Lakes or the Adirondacks. 65% of the people that were actually coming to our water bodies actually did report that they had been in a water body in the last 2 weeks. And although 20% are from a different water body, we are slightly in a different place because in Lake Ontario or the St. Lawrence, you could be 50 to 100 miles away from each other, but still technically in the same water body. So we tried to look at that regionally as well.
Hey there, people ask me all the time why I'm proud to be a watercraft inspection steward, and one of the biggest reasons I tell them is I am super proud to protect the waterways that I grew up on, that I recreate on, and I also have so many memories attached to them. And I want these waterways to be protected for years to come, and that helps with the prevention of spreading invasive species.
So we had the opportunity with some of the slower launch days and just a different perspective of what stewards thought would be more beneficial for them. So they all were talking about why they're proud to protect our waters. And so we actually were able to collect some videos and some awesome resources from our stewards about some of the reasons why they love this position and what they're doing. We're working on the report. So all of the data I just mentioned to you is actually considered to be preliminary. So those numbers will change. Working with Zach, our GIS specialist, to assist with creating and helping actually analyze and create new resources for our report. The 2021 launch profiles are being created by Zach, which they were so fantastically reviewed by everyone who has actually had access to them. So I'm excited to now be able to start initiating an annual trend at each launch so we can see the difference over each year or over time at the launches that we're covering. So super exciting information, and Zach is definitely working hard on getting that going. On top of all of the watercraft inspections our stewards are doing, we actually worked with, we had them out for 4,800 hours this year. We had 9 stewards, and we had them conduct rake tosses at 24 of the launches so that we can get a better idea of all the species that are found in the launch areas. We worked on special projects, including conducting a signage analysis. So figuring out what kind of signs were at all of our launches, what the quality of those signs were, if they had disposal stations, restrooms, things like that, so that we can start improving the signage and more of our messaging. We had stewards working together to collect photos, again, some of those videos, and then also creating captions for social media for more improved information sharing. And so if you follow SLELO on Facebook, you can see some of these updates on something Megan and I have coined as Water Wednesdays. So exciting. And if you're not following us on social media, you should. We had our stewards come out, and they actually developed a standardized operating procedure, SOP, for specimen collection and preservation. And this was something really exciting because we had a couple of the stewards to actually just take the reins on this, really find a bunch of really awesome resources for collection and preservation. And then we utilized that information. We went out onto our water bodies, collected, I think 28 different aquatic species. And the stewards got to help us press and preserve those species so that next year we can improve training and our stewards' knowledge and also their ability to share with the public. And I do want to thank Tilt and Save the River for assisting with the specimen collections as well. And then finally, just the last thing I just wanted to mention is that our program was co-administered with the Thousand Islands Land Trust successfully again this year. We're already looking forward to hopefully being able to continue that partnership into 2022 and ways that we can improve this program, change this program, and protect the waters in our region. So if you know anyone interested in being a steward or have any information you want to share about the steward program, please feel free to reach out. Next, I just wanted to just briefly mention the WIRL program, or the World Protectors Project WIRL, with the Indian River Lakes Conservancy. So we actually Robert and I met up with Megan and some students from WIRL to go out to Grass Lake, and we conducted a bunch of rake tosses, taught the students how to do rake tosses, how to ID native species, how to ID invasive species, just kind of talked about the importance of understanding both of these. And we went down to the Baker Island Preserve and did a hike on the island there, which is just a super unique and awesome actually ecosystem. And so it was really good for this partnership. We truly enjoyed doing that. And we also got to get an understanding of a lake that might not be a priority conservation area for us, but is a priority for some of our partners. So that was a very good, successful day. And if you haven't kayaked or paddled on Grass Lake, highly, highly recommend it. It was so beautiful. Moving forward to, I know Robert actually mentions our early detection field surveys this year, but I did find that I think that Whetstone Reservoir deserved an extra shout out. So Robert and I went out for a day and we conducted 16 rake tosses on the reservoir. We identified at least 18 native aquatic species, and we still have some unidentified species that we're trying to work through confirmation. And this included multiple native pondweeds, 3 species of bladderwort, water shields, betterdock, valsnaria, and just so many more species. And the reason why I wanted to give this special shout out is not a single aquatic invasive species was actually found in the reservoir. As you might know from past surveys, that we are in hopes and we're working with parks to submit an application to them to dedicate this as an invasive species prevention zone, or I.S.P. The only species of concern that we did find, and although it's kind of widespread throughout SLELO, was we did find creeping jenny found in a near a culvert that divides, there's a road that divides the reservoir. And so that was something a little bit of concern, just because creeping jenny isn't actually prolific on the Tug Hill region in general. So it might have been something that came in the soil in that spot. So that's something that will be in the report, and we'll share all that information. And again, special partner shout out to parks who sent 3 of their seasonal staff to climb into a canoe together and assist us with rake tosses. And Robert and I taught them about some of the different invasives and native plants while we were out surveying. So also, as you can see by these photos, it was the most perfect fall day that you can imagine. So very exciting day for us out there. Plus, when we actually don't see invasives, it's also kind of nice to be out on those days. So the next big project that I want to talk about is our aquatic restoration initiative. You might recall we worked with Roots last year to conduct a very in-depth survey about the native and aquatic plants, and then also looked at the riparian area. And those recommendations that came from that project were to it was actually most important to manage the Phragmites and Japanese knotweed in the riparian area there. So we successfully treated about 2.5 acres, and we are in process of removing all of the dead materials from those herbicide applications, and then also bringing in some native seed mixes to spread that are of species that are already found at that site. If you look at the top images on the slide, the one is on the left where, when possible, our subcontractor through Cardinal, they actually did a foliar herbicide application. But then if we were getting too close to the riparian or water areas, they just did stem injection. So a couple different methods, and a lot of time spent out there digging through all of that Japanese knotweed. And some of the photos on the bottom are some of the native species that we found at the site. We're in the process of creating an interpretive panel that is emphasizing restoration and the importance of native species, and then also why managing invasive species in riparian areas and why riparian areas are so important to our waterways. So we're going to try to match that similarly with some of the signs that are already there in that region. So we're very, very excited to see that come to fruition. And this is just a drone imagery of South Sandy Creek, which I actually worked with Zach Simek, again, our GIS analyst, to assist with this process. And it was just a very exciting piece of our work, because we are able to go from, if you look at where this small square is, we're able to go from a slightly blurred or poor quality spatial resolution from resources like Google Earth to just really totally enhancing this to the point where if somebody was honestly interested enough, you could probably count the total number of Phragmites stems in a stand. So pretty crazy. So this is what your Google Earth image looks like, but then this is what the drone imagery of that exact location looks like. So we are very, very excited to use this information and hopefully work with DEC again next year to be able to continue doing some drone mapping and start actually measuring the impact of our treatments at this site. And then hopefully we can use this project as just kind of the foundation to future restoration work and, moving away from just the consistent managing of invasive species messaging to that restoration and importance of native species for improving our climate as well. So as I'm going to start concluding here, I just wanted to share a brief video with you. I know we talk a lot about innovation and cool tools that SLELO has. There's no audio to this video, but we've been bringing out I have a GoPro, I've been bringing that out with us. We have the ROV, and we're starting to utilize different technologies that can help us get a better understanding of the species, the biological diversity, both the native species and invasives in our waters, just kind of all over in general, how they're interacting with each other. And this is a little bit of a learning curve for us. We're still learning how to operate the ROV. We have taken it out a few times now and just kind of trying to work on improving our surveys at our PCAs. So all super exciting. Hopefully you enjoy the different things that we'll pull together from the resources that we're using out in the waters. And, even thinking back to that video, it's so hard. People don't truly understand what's under the surface of the water. And without videos and good imagery and using technology, we can't truly get people to be as passionate about these areas as we are, because I can nerd out about plants all day, but not everybody wants to do the same thing. So we're excited to see what the future holds for that. This is my last slide before we move into our eDNA project. And so we're actually going to hear from Jacob. He is our eDNA project technician. He came on with us in August and will be with us through at least December. We're very excited to have Jacob on board. He's been coordinating all of our sampling efforts. He's been transporting our samples from our eDNA sampling to the lab that we're working with, and also working with our volunteers and partners in collaboration for this whole project. Jacob is another SUNY ESF graduate, like I know many of us actually on the line are. So he's another one from SUNY ESF, and he had extensive experiences in freshwater resources like fisheries and macroinvertebrates. So he was, and still is, I hope, very excited to get into the genetics world of eDNA. The funding from this project is through the Arconic Foundation, and the Arconic is actually, there's a factory up in Messina. And so they were really passionate about innovative tools in our waters because they are located right on the St. Lawrence River. So sorry to steal any of your thunder, Jacob. But last thing I wanted to mention is that our mobile lab that we've talked about a couple of times is actually currently in production, which is super, super exciting. And hopefully we'll have it built before the end of the year and then be able to start using it in 2022. So Jacob, if you want to go ahead and unmute, and I can change your slides for you if you just tell me next slide when you're ready.
Awesome. Yeah. So thank you, Brittney, for the introduction, and thank you to SLELO for giving me the opportunity to talk with everybody today. So as Brittney mentioned, I'm working on the tributary eDNA project. Next slide, please. Just as a little bit of a background for anybody that is unfamiliar with eDNA, this is just any genetic material that is given off by an organism into its respective environment. So this can be soil, it can be water, it can even be air for that matter. So it's pretty cool. And tagging off of what Brittney was saying before, there's a lot of great tools that are coming out that are allowing us to do really, really cool work. So it's proving to be a cost-effective tool for biological work, and there's still a lot of research that is being done and a lot of other avenues that are being explored for utilizing eDNA. As it pertains to this particular project, we are utilizing eDNA for targeted invasive and native species, and considering this is a PRISM call, we have the invasives listed right there. So Asian carp, tube-nosed goby, northern snakehead, tench for the fishes, and then we have rusty crayfish as our invertebrate. Next slide, please. So pertaining to why we're doing this in tributaries, there has been some research that has been coming out in the last few years on other Great Lakes showing that corriganines or whitefishes are utilizing tributaries for their spawning behavior. They're commonly known to spawn in lakes, and so this is an interesting find. And that begs the question with our Lake Ontario whitefishes, are they also using tributaries? So it'll be interesting once results trickle in to see if we get any hits for that. And for the invasive species, it's still furthering the understanding of possible distributions. Hopefully there's a lot of non-detections for invasive species, but if they are there, this will be really great to get a better idea of that distribution. Next slide, please. And so going into some more details pertaining to the actual project, Brittney and Pippa went through the process of identifying 15 tributaries using aerial imagery to go out and sample, and that spans all the way from Oswego up into Messina. So we do have a really great spatial spread. And it was part of my job to go out and then verify on the ground that we had sites to sample long-term, so throughout the duration of the project. And as you can see, there's several parameters that we were considering. Accessibility was one of the bigger ones, especially once we get into December and possibly snow and all that fun stuff. But we were also looking into things like flow, things like substrate, and I'll talk more about the ROV later, but there will be some interesting, hopefully some interesting work that we can do later on to get more granular views of these types of parameters. Next slide, please. And so just going along with TNC and SLELO's ties to volunteer work, we held a volunteer event up in Messina at the Nicandry Nature Center. This was back in September. It was a really great day, as you can see with the image on the left there. It was a beautiful sunny day. It was great to interact with everybody, and we basically just went through the process of showing people how to collect samples, filter them, and preserve them, and then chit chat about a variety of the other nuances pertaining to the overall project. So it was a great day for sure, and thank you to the Nature Center for having us. Next slide, please. As for the laboratory work, we're partnering with Dr. Nicholas Sard at SUNY Oswego. He does a lot of eDNA work himself. He's very knowledgeable. I've been pretty lucky to chit chat with him over the last month and a half, so that's been really cool. He will be using qPCR to run the analyses, so that's just quantitative polymerase chain reaction. Say that 10 times fast. The most current update that I have is the materials are still in the process of being synthesized, so we don't have any results for any sampling we've done now, but hopefully within the next few weeks, those results will start to trickle in. Next slide, please. As for the actual sampling update, so of the 15 originally identified tributaries, we were able to do all 14 in September. That amounted to 56 total samples. October sampling is still ongoing, and I was actually sampling Monday through Wednesday of this week and was able to get a lot done, so that's been great. And it's likely we'll be done with October next week with all that sampling. And the reason why 14 tribs instead of 15, we're still in the process of communicating with the Akwesasne tribe to gain access to the St. Regis River and get some samples there. And so hopefully soon we'll be able to sample there as well. It's a great river and it's a beautiful river. So hopefully we'll be able to get some samples from there. We have been pretty fortunate to have some volunteers and partners come out with us for sampling. So it's been really great to communicate with them and just have fun, and the beautiful weather that we have been blessed with. It's looking like it's going to turn coming up here, but you know, good things can't last forever, right? Next slide, please. And yeah, so relating to the ROV, this is our remotely operated vehicle. We were able to take it out onto North Sandy Pond. And so the images on the right there, at the bottom, that's myself and Robert trying to figure out how to work with it. And so we're still working out the kinks of utilizing it, but hopefully as we learn more about it and how to operate it, we'll be able to collect some more data that way. And so, yeah, it's definitely very useful for observing habitat and potential species in the area. So I'm a little biased, but I'm pretty excited to continue to work with it and see what kinds of data we can get from that. Next slide, please. And I just wanted to give a shout out to our volunteers and partners. So New York State Parks has been very, very helpful. They were actually out with me yesterday and Tuesday, and it was a lot of help. I think I saw Peter in the chat earlier, but shout out to Peter Zimmer and Elizabeth Snow. They've been awesome. Again, shout out to McAndrew Nature Center and their staff up there. It's a beautiful spot. And Joel Danko, who is their trail manager, he's been very helpful to us as well with getting more sites up in the Messina area. So providing some good details for some solid spots to go and sample. So that's been very, very helpful. And to all of our volunteers that have come out so far, thank you. It's been great. And I look forward to continue sampling with all of you and learning more about what you do and doing all that fun stuff. And yeah, so again, just thank you everybody for letting me speak today. And also with the volunteer shout out, we're always open for more people to come out and sample with us. So if you're interested, you can email me directly, or you can go to the SLELO website and fill out the form. And yeah, and I believe Brittney can correct me, we might be having volunteers out for working with the ROV, so there might be some more opportunities coming up in the future. But yeah, if anybody's interested, it would be great to chat with you and hopefully work with you in the future. Thank you.
Hey, thank you, Jacob. I just love this eDNA stuff. It's just something very, very intriguing about it. I think what we'll do next is hold off again on questions until the end of the meeting, and we can have a kind of an open dialogue. And next we'll hear from Megan on education and outreach activities.
Hey, everybody. Nice to hear your voices. So I'm just going to give you a snapshot of stats for this quarter. So we've had 78 Facebook posts. We've reached 9,863 people and engaged 10,670 people. We've also hosted 10 events, and we've engaged 179 people at those events, and we've had over 10,000 hits on our website. And if any partners are out there who are organizing water chestnut polls and you need assistance getting their treatment data into IMAP, please do reach out to me. I can assist with this. The reason why we're really interested in, well, one of the reasons why we're interested in getting this data is because IMAP has developed an application that will utilize the data to generate a visual dashboard that's going to capture the effectiveness of water chestnut pools over time. And the deadline to have that data into IMAP is the beginning of November. So we're just reaching out to partners to try to get that data into IMAP. I will place a link to a step-by-step guide on how to report your water chestnut data into IMAP shortly after my presentation. So the Pledge to Protect has launched. You've been hearing about it for a while now. So the Pledge to Protect is a fun, engaging, and rewarding way to participate in invasive species prevention and management. The program is designed to give pledgers the tools and simple actions they can take every day to make a big impact in protecting their outdoor spaces, forests, trails, waters, backyards, and community. So far, we have over 30 pledgers, and we're encouraging our partners to take the pledge too. And I was going to just give a quick little tour of the Pledge to Protect landing page. You should be seeing my screen now. And this is where it lives. You go to ipledgetoprotect.org. It brings you to this page here.
I don't believe we're seeing your new screen.
Oh, you're not? Interesting. Let me try again. Is it working now?
Not at my end.
Why is it being pesky?
Rob would have to stop sharing for you to start sharing, Megan.
How about now? Yes, no, maybe.
Yes. Yeah.
Yes, awesome. Okay, so you go to ipledgetoprotect.org. It brings you to the landing page that hopefully you are seeing now. The pledge category that you choose down here, this dictates the type of information that you receive in the monthly emails. So for example, if you choose to protect lands and trails, you will receive monthly emails that showcase simple actions you can take to protect your favorite hiking trails and lands. Whereas if you choose to protect them all, you'll receive a monthly email that showcases highlights from each pledge category itself. Pledgers, they receive monthly emails showcasing the simple actions that you can take to fulfill your pledge. You also get bragging rights, collect badges, and win prizes for completing action items outlined in your monthly emails, and access to a cool social media toolbox, which you can see here on the screen, and chances to win cool swaggy prizes, which I have some here. We've got a window cling, we've got patches, we've got cool magnifying, light up magnifiers for searching for HWA, and awesome cell phone cases for water protectors, and more stuff on the way. And there's virtual toolboxes. You can look at a plethora of resources here, and it's a pretty cool site. So I'm encouraging all of our partners to take the pledge. And Rob, you can go back to sharing your screen for the slides, please and thank you. Awesome. Okay. Can you go to the next slide for me? So moving on, so Rob mentioned this earlier, salmon season is bringing in many anglers from known spotted lanternfly infestations. We got people coming from across New York State and beyond. So we visited some fishing hotspots along the Salmon River and spoke with anglers to inform them that they could unknowingly have SLF on their vehicles or their equipment. We used our trackable brochures through the SLF Spotters program, and we handed them out to individuals as well as dropped off in bulk to popular tackle and bait shops in the region, and also visited the Douglas's Salmon Run and the Altmar Fish Hatchery and gave out these brochures. And people were really, they received this information well. A lot of them already knew what SLF was and were really excited to learn the simple ways that they can help stop the spread of the insect just by checking their vehicles. And they agreed to go ahead and scan the trackable QR code and take the survey that we have put together through the SLF Spotters program. So I'm looking forward to reviewing that information and seeing some stats from that. Next slide, please. And as always, you can visit our website to learn of upcoming events. Here's a couple that are coming up that you might have interest in attending. So also I'd like to mention, you can go ahead and click for the animation there, it should come up. Okay, so I'd like to mention that SLELO gained international recognition at the North American Invasive Species Management Association Conference that was held this past September. Team SLELO presented on high-level topics that explored the importance of invasive species management and conservation work and enhancing the resiliency of transboundary landscapes through small-scale invasive species management initiatives. Next, please. All right, so the DEC is premiering a new invasive species documentary titled Uninvited. The documentary introduces the concepts of invasive species and highlights some of the species threatening New York's environment and economy, while also showing some innovative ways that New York State is combating these threats. Uninvited also features the collaborative work of the DEC and its partners, including New York State Department of Agriculture and Markets, PRISMS, and New York Invasive Species Research Institute, and many others. Rob, you can go ahead and play this trailer. And just a reminder, you have to have that button clicked.
Invasive species can be spun as a conservation issue, as an ecological problem that only the scientists or the nature lovers care about. But it actually does impact our daily lives.
I used to have a back lawn back here.
It was coming this way towards the foundation of the house.
Giant hogweed contains skin-burning sap.
As humans, we tend to dominate ecosystems. We bring species around, we enjoy them. That gives us incredible power.
Because since when is extinction the only measure of a problem? That's like only going to a doctor when you're dead. You know, one of the issues we have is that we have stopped thinking of ourselves as part of the natural world. We're something separate. And we've fallen into the trap of thinking we don't need the natural world.
So we're looking for spotted lanternfly, particularly the egg masses.
Surveying for southern pine beetle.
Hemlock Adelgid.
But your clean water, your air, your food, moderate weather systems, all of these things are produced by functioning ecosystems. They'll do more poorly when we take those species away. And that's what invasives are doing. They're reducing the number of species in our local ecosystems.
Invasive species becomes that issue that we just want to throw up our hands and say, there's nothing we can do. I know it seems like we have a lot now, but there's a lot more species that could be introduced if we don't act now.
Okay, so I get lots of cold chills every time I watch that. And if anybody is interested in hosting a viewing, you can do a virtual viewing, you can do an in-person viewing, whatever you choose. I put a link in the chat for you to go ahead and register or show your interest to do that so you can get the toolkit that you'll need to go ahead and host a viewing. Thanks for listening.
Super. Thank you so much, Megan. So I think at this point, I know there's been some questions coming up in the chat box. And if those who presented today are still on the line, what I can do is I can go through the chat box and pull out some questions. And I think as we go further back into the chat box, we had a question. I think this is directed to Robert and Brittney about where the yellow iris was found, what priority conservation area that was found in. So if any of you could help with that, that would be great.
Yeah, the yellow iris was identified last year at Monterey Point boat launch, where one of our stewards was stationed. So that's where that was.
Super. We had another question. I think this is for Robert about the Jacob asked what was driving the differences between cage 1 and cage 2 with the Hypena opulenta. Why were we seeing different results in the different cages? Robert, if you could comment on that, that'd be great.
Yeah, that's a tough one because you know, last year I was starting to believe that the amount of sun that the cages got was making a difference because Grenadier Island and Carlton Island were out in open areas where we had the cages set up last year, while Whaley State Park was in a forested area, and Whaley did really well, and we got nothing from the islands. That, you know, didn't hold true this year since We've set up 2 on Grenadier Island, and they did better than the ones in the forest area in Whaley, and only one of the Grenadier Island cages was in the forested area. So I think there's really too many variables going on to say for sure why the one did better than the other.
Super, thank you. And Irene asked the question about copies of field reports. Now's the time of the year when we start putting those together. Once we have them completed, we post them to our website, and anyone who would like a link or a copy sent direct, we can do that upon request. Our annual report usually comes out in January. We have a contractual obligation to get that out early January. So, feel free to contact us, and as those reports are finalized, we will make them available. Let's see, other questions. Just searching through the chat box here. Megan, have I missed any? Do you know?
Irene asked if the footage from the drones was being shared with DEC.
Yeah, that's a good question, Megan. The answer is yes. We just started putting the drone footage together. So Irene or whoever else, I know Irene, you're leaving us soon, but whoever wants it, I can definitely share that with you.
Great, thank you. So if you have any other comments or would like to make an announcement, feel free to put that in the chat box. Megan, if you could just monitor those for me, that would be great. And I'd like to circle back to see if Sarah Trick is on the line from Indian River Lakes Conservancy. I know that she wanted to make an announcement. Are you with us, Sarah?
Yeah, sorry about that earlier. Time got away from me this morning.
The podium is yours.
Okay, so we just wanted to thank you guys for working with us on our world program. I saw that mentioned in the thing, but we had a great time out on the lakes and hiking with you guys. And then we're really excited to work on the water chestnuts threatening Black Lake. And we were just hoping that maybe somebody on the line would know of any grants to help fight Phragmites, because we found them on one of our preserves and we're trying to figure out how to deal with them monetarily.
Sure, I would say that if anyone has ideas for Sarah, feel free to email her outside of the meeting. And I think, when it comes to Phragmites, that we're all kind of wishing and hoping that that biological control will be effective and be approved and able to start field trials. Is that it, Sarah?
Yep.
Super.
Hey, Rob or Sarah,
Yes.
I'm going to put in the chat here, Sarah, a link to grant opportunities document that Emily Fell put together a while back. So I'll send that your way and you can look over that and see about grant opportunities.
Thank you.
Super. So I would like to, again, thank all of our partners and our cooperating affiliates, for your patience and understanding again this year. This whole pandemic thing has altered the way in which we work, and we're grateful for everyone's commitment to invasive species prevention and management and to our PRISM. I also wanted to thank and acknowledge our team, Robert Smith, Megan, Brittney, and Zach for the major contributions towards our program and mission. Nice work, team. Our next meeting is scheduled for Wednesday, January 19th. We hope everyone can attend. Please note that our meeting dates for 2022 are shown here. I'd like to put those out as far in advance as possible, recognizing that, there may be times when we have to shuffle a date due to some other major event or for whatever reason. Our offices at the Nature Conservancy unfortunately remain closed at least through the first part of January. Our organization is being, very careful and whatnot as we, move to reopen. We hope we're going to reopen this fall. Looks like that's been pushed off at least until January. That said, we are always available. You'll see our email addresses here. Feel free to reach out to us at any time. And this brings us to any last minute discussion that anyone would like to have or any announcements you'd like to make before we close.
Hey,
Hey,
Rob,
Robin.
you have 2021 on the dates. You mean
Yep.
2022?
Yep. So my apology. That would be next year.
Oh,
So all the 2021 dates are next, it actually should be 2022.
oh, going along that same theme, Rob, I was going back to my management numbers for slide 1. And the correct number for swallowwort acreage treated this year is 69.5.
Super. Thank you, Robert.
You're welcome.
Anything else from anyone?
I want to just thank you for everything you guys do, and I enjoy working with you as a GEC employee. I do plan on being still active some as a citizen, so I will give you my home email, and hopefully I can keep involved.
Super. Well, thank you, Irene, for everything that you've done back in, helping to pioneer our PRISM, if you will, and to get the PRISM formally underway, and also everything that you've done to help protect our lands and waters from invasive species and other threats. Good luck with your retirement. We wish you the best and hope to see you in the future. Great. Well, with that, thanks again, everyone, for taking time out of your busy schedules to be with us today. Again, if you have any other questions that come up, feel free to reach out to any of us. We'll do our best to be responsive. And with that, we'll wish everyone happy holidays. And thanks again for being with us today. Be well, do good work, and talk with everyone soon. Thank you.